This mode is accessible when the kidneys are selected from the main menu. On the left, one can see an icon for the excretory system, kidneys, bladder, and blood vessels that feed the kidneys. Generation of urine in the bladder proceeds in real-time scale. In order to view full information on the biochemical composition of urine, left-click on the line called urine highlighted in blue and located under the bladder icon. Options nephron right and nephron left allow to observe in real time the uropoiesis mechanism in the left and right kidney accordingly. When you left-click any of these icons, an interactive image will appear. It demonstrates a nephron, a functional element of uropoiesis consisting of the glomerulus, a tangle of vessels where the primary urine is filtered, the loop of Henle, and the collecting duct. Directly next to the glomerulus icon, one can find indicators displaying readings of incoming and outgoing hydrostatic blood pressure, as well as the pressure of urine in the descending limb of the nephron. Along the entire loop, one can see indicators of osmolar reabsorption, the osmotic gradients that lead to the second phase of uropoiesis. On the right, one can see 3 graphs. The top and the middle graphs display the dynamics of hormone levels in the blood, regulating the second phase of uropoiesis. The bottom graph displays diuresis. Let's go back to the main menu of this mode. In the top center of the screen, there is a test tube icon containing the total amount of blood in the organism. Specific numerical values of blood in milliliters and the hematocrit levels can be found above and to the left of the test tube icon respectively. At the top right corner of the main menu, there are 3 rectangular columns with dynamically changing content, determining, from left to right, the total water content in the blood of the organism, water content in the extracellular environment of the entire organism, and water content in the intracellular environment of the entire organism. Specific numerical values of water in milliliters are indicated under each column. Inside the columns, at the top, the corresponding values of the osmotic concentration of substances in water are displayed. In order to obtain full information on the biochemical composition of substances in the blood, the composition of substances in the extracellular environment, and the composition of substances in the intracellular environment of the organism, left-click on the blood, extracell, intracell lines respectively, highlighted in blue and located under the rectangular columns. Let us demonstrate what happens if we choose to see the biochemical composition of the blood. To get readings in terms of concentration, left-click on the appropriate millimole per liter lines, also highlighted in blue but located a line lower. To sum up the total biochemical composition of all substances in the organism, blood plus extracellular environment plus intracellular environment, left-click the sum line highlighted in blue. Finally, to see the biochemical composition of the blood cells, left-click the blue cells line. At the bottom right corner of the main menu, there are rectangular columns of a smaller size with dynamically changing content, determining the water content in the blood of individual organs, the extracellular water content of individual organs, and the intracellular water content of individual organs. Similarly, specific numerical values of water in milliliters are displayed under their respective columns, while the osmotic concentration of substances in the water is provided inside the columns at the top.